Hello, and welcome to Back to the Science. I'm Dr. Susan Oliver, and I'm a scientist, and this is Cindy Oliver, and she's a dog. As most of you will know, the 2023 Nobel Prize in Physiology or Medicine was recently awarded to doctors Katalin, Karikó, and Weissman for their discoveries concerning nucleoside base modifications that enabled the development of effective mRNA vaccines against COVID. Now, most scientists who have knowledge of the field think the award was well deserved. But there is one scientist who is a little bit miffed, and that is Dr. Robert Malone. This morning I was telling Cindy about this, and then when we went on our walk, Cindy picked up this dummy and carried it home. I think she was trying to tell me I should change the title of the video to something about Malone spitting the dummy. But I thought I should check whether everyone actually knows what the term means first. And it turns out they don't. So we won't be doing that. But it was a good idea from Cindy, nevertheless. And maybe we could still put it into the thumbnail. Anyway, this is what Malone posted on the platform formerly known as Twitter. Karikó and Weissman get the Nobel, not for inventing mRNA vaccines, because I did that, but for adding the pseudouridine that allowed unlimited spike toxins to be manufactured in what could have been a safe and effective vaccine platform, if safely developed. Good to know. So he's not too impressed. Some may say he has sour grapes. And if you know what it means, you might say he has spit the dummy. Anyway, he has made two claims here. Firstly, that he invented mRNA vaccines, and secondly, that pseudouridine allows unlimited spike toxins to be manufactured. So, in this video, we will look at both claims, starting with the claim that he invented mRNA vaccines. But before we do that, let's look at how long he has been making this claim. If we use the Wayback machine to go back to his website in September 2020, before mRNA vaccines were approved for use, we see that he makes no mention of mRNA vaccines. But he does claim that he is known as one of the original inventors of DNA vaccination. Fast forward to May 2021, after the mRNA vaccines were approved, and he is now the inventor of mRNA vaccines. Go forward a little further, and he didn't just invent mRNA vaccines, he also invented the whole field of mRNA therapeutics. Wow! And if you listen to his fans, he did even more. He actually invented RNA. That one is, of course, very easy to debunk because RNA first appeared on Earth well over 1 billion years ago. But what about his more modest claim of inventing mRNA vaccines? This figure here shows the various scientific advances that were key to the development of mRNA vaccines. Malone was responsible for one of them, which is this one here that I have put a box around. But there were a lot of important things that happened before and after his input, all of which were important. Among the things he didn't do, he didn't discover mRNA. He wasn't the first person to produce proteins from isolated mRNA in a lab. He didn't develop lipid-based delivery systems. He wasn't the first person to deliver mRNA to cells and translate it into protein. And he wasn't the first person to synthesize mRNA. The people responsible for developing the technique to synthesize mRNA were doctors Paul Krieg and Douglas Melton, who, in addition to developing the synthesis technique, also determined that it was necessary to cap the mRNA in order for it to be translated into protein. But Malone did do something. He used a technique developed by Krieg and Melton to synthesize capped mRNA, which he then encapsulated with a cationic liposome and subsequently delivered into a range of cells. And he showed that these cells translated the mRNA into protein. And he also confirmed what Krieg and Melton had previously determined regarding the necessity of capping the mRNA. Now, you may be wondering where the cationic liposomes that he used for the encapsulation of the mRNA came from. They were supplied by Dr. Philip Feldner, who had previously developed them and then used them for transfection of DNA. Feldner had done some initial work encapsulating mRNA with his cationic liposomes, but hadn't been as successful as Malone, as his mRNA wasn't capped and he also wasn't using a sensitive enough detection method for the protein produced. So, Malone's research was an important step in the journey towards developing mRNA vaccines, but he was hardly the inventor of them. And there were more important steps before a viable mRNA vaccine was developed. One of them is, of course, the subject of the Nobel Prize. But before we get to that, I would like to talk about another important scientific advancement, which didn't get a Nobel Prize, but did get the scientist responsible a number of other awards. And that scientist is Professor Peter Kullis. And what he did is he developed the lipid nanoparticles that are used in the mRNA vaccines. What's special about these nanoparticles is that they contain an ionizable cationic lipid. This lipid is neutral when it's in the blood, but when it enters the more acidic endosome of the cell, it gains a proton or hydrogen ion and becomes positively charged. And as a result, the nanoparticle releases its payload of mRNA, which is pretty cool. And speaking of mRNA, that's where doctors Katalin, Karikó, and Drew Weissman came in. A key issue with the mRNA that was initially synthesized was that it caused major inflammatory reactions. This both increased unwanted adverse reactions as well as limiting the amount of protein that could be produced. So Karikó and Weissman decided to investigate. And initially, they noted that RNA from mammalian cells frequently contained a modified base known as pseudouridine, whereas synthesized mRNA instead contained uridine. They wondered if the absence of modified bases in the synthesized RNA could explain the unwanted inflammatory reaction. To investigate this, they produced different variants of mRNA, each with unique chemical alterations in their bases, which they delivered to dendritic cells. And they found that the inflammatory response was almost abolished when base modifications were included in the mRNA. And what's more, they also demonstrated that pseudouridine containing mRNA was more efficiently translated, resulting in higher protein production in cells that had taken up the mRNA. So, by using these base modifications to both reduce inflammatory responses and increase protein production, Karikó and Weissman eliminated critical obstacles which were in the way of using mRNA for clinical applications. And that's why they got the Nobel Prize in Physiology or Medicine. But what about Malone's claim that the modifications allowed unlimited spike proteins to be manufactured? Well, we can easily tell that this isn't true by going back to the studies originally performed by Karikó, Weissman, and colleagues. So, in this study, in addition to testing cultured cells, they also encapsulated mRNA designed to express luciferase into liposomes and injected them into mice. Luciferase is bioluminescent, so by doing this, they are able to use imaging equipment to see where in the body mRNA is expressing luciferase. So, firstly, they injected the modified mRNA and tested for luciferase activity in a number of organs. And note, this is intravenous injection. not the intramuscular injection that is usually used with vaccination. And what they found was that there was high luciferase activity in the spleen, but not in the lung, liver, heart, kidney, or brain. What they then did is they compared the luciferase activity of modified versus unmodified mRNA in the spleen over time. And what they found was that the modified mRNA did produce significantly more luciferase than unmodified mRNA, but the production wasn't unlimited. It had already substantially declined after 24 hours. And importantly, pro inflammatory cytokine levels were significantly lower in mice injected with modified mRNA. So the modified mRNA was doing exactly what it was designed to do, producing more protein with less inflammation, but not forever. So Malone is wrong when he says he invented mRNA vaccines, and he is wrong when he says they produce unlimited spike protein or spike toxins, as he calls it, just to make it sound more scary. But enough about him. Let's finish on a positive note. Nobel Prize winner Katalin Karikó was demoted 4 times at her old job, but she never gave up. She persisted by focusing on what was next. Something to remember when things initially don't go according to plan. If you'd like to look further into the data I've presented, I've provided links in the video's description. And please remember, this video is about the science, but you shouldn't take it as medical advice. For that, you should speak to your medical practitioner. If you've got this far, thank you for listening. And if you've liked, shared, or commented on the video, double thank you, because that helps the algorithm. It means that more people will see the video. And of course, thank you to everyone who has bought me a coffee or very clever Cindy here a treat. So clever coming up with the dummy idea, even if it's not that suitable for the title. But we really appreciate your support. We will be continuing to make videos about the science in the future. So if you'd like to see them, please hit the subscribe button. Thank you.